everyone, welcome back to our unit on cellular respiration. So in the last few videos, we covered the reactions of cellular respiration, including redox reactions and the synthesis of ATP. Then we also looked specifically at glycolysis, pyruvate processing, and the citric acid cycle. In those previous processes, we were able to gain some redox potential and a little bit of ATP, but today we're going to look at that big payoff, how we get that 32 to 36 ATP that you always see in textbooks. We're going to be exploring this through the electron transport chain. So let's again recap what we actually had as we finished those last processes. So we did start glycolysis, remembering that this is in the cytosol of eukaryotic cells. We broke down the 6-carbon molecule into 2 3-carbon pyruvates, which was then transported into the mitochondria. The citric acid cycle happens in the matrix of the mitochondria, and we're able to again generate some redox potential and some ATP. So again, today we're going to be finishing up our cellular respiration with electron transport. So our main cellular respiration reaction, if we remember, this is very similar to a combustion reaction. So we have glucose, which is going to be, again, our 6-carbon molecule, and we can add oxygen and get water, carbon dioxide, and energy. So again, the difference between this and a standard combustion reaction is that we don't allow the oxidation of glucose, these carbons, we don't allow those electrons to snap quickly to oxygen. Instead, what we're going to do is have this travel through progressive stepwise oxidation, glycolysis, citric acid cycle, so that we slowly allow these electrons to go to oxygen. So far, in our previous videos, we have, of course, seen that glucose is an input, so we can check that off. We've seen where our carbon dioxide comes from. So again, 2 of these carbons left as carbon dioxide per glucose during pyruvate processing. An additional 4 carbons left per glucose from the citric acid cycle. So again, this is the carbon dioxide that you exhale. This is all the product of cellular respiration happening. So again, check that off. We haven't really talked about oxygen, where that's actually going to come into play, and we haven't talked too much about water, but we'll see a little bit about that at the end of this too. As for energy, we still have a few ATP. Remember, we did get some from glycolysis and the citric acid cycle, but we're still waiting on that 32 to 36 ATP payout that everybody talks about. So what exactly did we get? In glycolysis, again, we broke down our glucose into 2 molecules of pyruvate, and this gave us 2 ATP, net ATP, and 2 NADH. Remember that NADH is important. That's our electron carrier. That's where those electrons go before they can make it to oxygen. After that, we process pyruvate just as we were going to enter the mitochondria, just after we enter this, and we're going to end up with 2 NADH per glucose. Okay, the citric acid cycle, we get an additional 6 NADH, 2 FADH2, which is an alternative electron carrier, and 2 ATP. So this is going to give us combined 4 ATP. Remember, these are those sort of bonus fry ATP, so we're not at the payoff yet. 10 NADH, which we'll be using today, and 2 FADH2. So these are the really important payouts that we've gotten from all of the processes we've talked about up until now. So again, glycolysis happens in the cytosol. Our citric acid cycle was happening inside the matrix, this sort of inner space of our mitochondria. So electron transport, we're going to actually be kind of staring at this junction right between the inner membrane here, the inside membrane, and the outer membrane. So we're going to be paying a lot of attention to this inner membrane and the space between the inner and outer membranes. This is going to be our inner membrane space. So we're going to be playing around a lot here today. So on the next slide, we're going to start to become acquainted with the complexes of electron transport. And so again, what we're going to be looking at is going to be this inner membrane. We're going to be situated right here for the majority of what's going on today. Okay, so here is our inner membrane, so it's that phospholipid bilayer. Here is the matrix of the mitochondria. So again, that's going to be just that inside central space. Here is the intermembrane space, and then somewhere even up further, not pictured, it would be your outer membrane. So that's where we're hanging out. So we're kind of in that sort of cristae infolding area. Okay, so you can see that we have some stuff going on here, and it might look really complex, but we're going to address them kind of one by one. So the electron transport chain consists of 4 major complexes, numbered 1, 2, 3, and 4, and 2 mobile electron carriers. For now, we're calling them Q and site C. Okay. As electrons travel through these complexes, which we'll get to in a minute, they'll build up a concentration gradient of protons that we'll be able to use through ATP synthase. So that's kind of the overall picture of what we'll be looking at, but again, we'll get to each of these complexes on their own. So let's of course start at complex 1. So complex 1 is also referred to as NADH dehydrogenase. So probably you can guess what this is about to do. The NADH dehydrogenase complex, or alternatively complex 1, is the drop-off point for electrons being ferried from glycolysis, pyruvate processing, and a citric acid cycle, specifically in the form of NADH. This complex contains 2 main redox sites. One is called flavin mononucleotide, FMN here. The other is an iron sulfur center. So these kind of look scary because we've not really talked about anything like this before, but you don't have to think too hard about it. We'll kind of come back to these at the end, but these are going to be the specific locations on this complex. It's a pretty large complex relative to an electron. These are going to be the specific places that will hold our electrons as they pass through, being temporarily reduced and then oxidized again as an electron leaves. So these are just the specific sites. So I want you guys to kind of loosely think about these because again, I'll bring them back up at the end for a recap. For now, think of these as kind of like the hands of the complex that are able to hold electrons. So they'll just be exactly where those electrons can land. Okay, so we have our complex 1 kind of set up. We know that it is about to receive electrons. So before we actually give this electrons, I think it's important just to review NADH for a second. So NADH is formed when 2 hydrogen atoms are taken from carbon. Remember, dehydrogenase, we're taking hydrogens. So remembering also that our hydrogens consist of 1 proton and 1 electron. That's it, pretty much. Hydrogen, 1 proton, 1 electron. Okay, so when we actually form NADH, we're given NAD+, which is the oxidized form, NAD+. What we're actually holding, again, is kind of 1.5 hydrogens if you think of them in terms of protons and electrons. So we'll have 1 electron. Think about that as kind of neutralizing that charge. Yes, there's a proton here. Then 1 whole hydrogen. So yeah, there's a proton left behind. We don't really have to think about that right now. Protons can do a lot of different things, but for now, don't worry about it. All we care about is that we're holding 2 electrons, and it might be worth noting that you've kind of got a potential proton here for the future. Okay, so now we know what NADH is actually holding. Let's go back to complex 1. Here's our NADH. We know that secretly there are 2 electrons here. So electrons will be passed first from NADH to our flavin mononucleotide part of complex 1. So as we do that, the electrons are passed. Remembering that we had that sort of spare proton hanging out with our electrons, we also have NAD+. So our NADH is oxidized to NAD+, and the proton's left behind, but we don't have to worry about it yet. We also see that the electrons are safely situated for right now on this flavin mononucleotide. So at this point, NAD+ can return to collect more electrons from earlier steps. We can see it maybe again later. And electrons are passed from the flavin mononucleotide to the next place where they go in this complex, the iron sulfur center. So we're still within complex 1. These are just 2 locations on this complex. You may also notice that there's now kind of a population of protons down here. The movement of the electrons through this complex causes protons that are lurking in the matrix to be pumped from the matrix into the intermembrane space. This will be how we're building that concentration gradient. For every 2 electrons from NADH that pass through this complex, 4 protons will be pumped. We will come back to this. You don't have to remember it in your head, but just kind of keep a relative idea of this, and we'll get back to the 4 protons. For now, let's go back to where our electrons will end up. So they're finished at complex 1, and they're ready to move on. So their next step will be to a molecule that we call ubiquinone, or alternatively coenzyme Q. And this molecule, ubiquinone, is a little bit different than the complex. So ubiquinone is going to be a mobile carrier. So it can actually, it's hydrophobic and can freely diffuse in the fatty acid tail part of this phospholipid bilayer. So within the inner membrane, it can just diffuse back and forth. So its job is to ferry these electrons between complexes 1 and 3. So we'll be skipping 2 for now, but we'll come back to it. So the electrons onto ubiquinone they go, and over to our complex 3. So the next stop again is going to be complex 3. This is also called the cytochrome B-C1 complex. And you can see that we've got cytochrome B, cytochrome C1, so that makes sense. This complex has 3 main redox sites. So you may recognize, again, an iron sulfur center. This is a very common type of way or place to hold electrons within these complexes. But now we've got some new things, cytochrome B and cytochrome C1. Cytochromes are very common within these complexes. We'll see some more when we get to complex 4, and when we get to chapter 10 and talk about photosynthesis, we'll see some cytochromes again. So these are very common sites where electrons can be held in these complexes. So cytochrome B, an iron sulfur center, and cytochrome C1. Okay. So for now, what we're going to see is that electrons will pass through each of these sites within complex 3. So here they go, cytochrome B. Ubiquinone can just go right backquinone to pick up some more electrons from complex 1. So we don't need to worry about it right now. So let's get back to these electrons. They'll pass to the iron sulfur center and then to cytochrome C1. So got some protons down here, a little bit of foreshadowing. Just like with complex 1, this flow of electrons through this complex will be used to pump protons, again, from the matrix to the intermembrane space. So there they go, out into the intermembrane space. Again, 4 protons are pumped here. This will give us a total of 8 so far. This will be important later when we're calculating our ATP payoff. Okay, so we're finished at complex 3. So the next thing is to go to our second mobile electron carrier called cytochrome C. So cytochrome C actually lives within the intermembrane space, but it's kind of loosely affiliated with the inner membrane. So it can kind of diffuse freely, but it's sort of skating along this inner membrane. So this type of carrier can only hold 1 electron at a time. So we'll put one on it, and we'll send a second molecule to get our second electron. We'll take these over to complex 4. Both of our cytochrome C's arriving. Complex 4. This is also called cytochrome C oxidase. So I bet you can guess why this would be called cytochrome C oxidase. About to just take some electrons from the cytochrome C. So we've got a couple of redox sites here. Cytochrome A, cytochrome A3. Okay, so cytochrome A, cytochrome A3, electrons, cytochrome C. We'll head back to complex 3. We don't have to worry about that. Finally, to cytochrome A3. Here, we can see that there are some protons lurking. At this particular complex, we only pump 2 across. Okay, so that's fine. This is a net 10 protons from complexes 1, 3, and 4 from the NADH that we put in. So we're kind of keeping a tally. You don't have to memorize it. We'll come back to it later, but just so that you get an idea of where this comes from. So something really interesting is about to happen at complex 4. So let's kind of zoom in on this guy. We're, again, looking mostly at the matrix space down here. So complex 4 is also the end of the line for these electrons. This is the step where they're about to finally go to oxygen. So 4 videos later, we're finally about to reach oxygen. So in order to complete this, what we'll need is going to be our 2 electrons, 2 protons. These are not the ones that got pumped. They're just down here. There's a lot of protons down here. And an oxygen. So this is called 1/2 and O2, because O2 is the natural, this is molecular oxygen, this is the way that it exists. So the correct way to really write this is 1/2 and O2, but this is basically, at the end of the day, going to just give us an oxygen. Okay, so let's combine our ingredients. All right, everything combined. This makes water. What? How did that make water? What are we even talking about? Let's take a look at this a little bit more closely. So here are our ingredients. We had our 2 electrons, our 2 protons, and a 1/2 and O2, aka an oxygen. So one thing that's helpful here is to remember, again, that a hydrogen is 1 proton and 1 electron. So let's go ahead and put those together. So what we really have here is going to be 2 hydrogens. So let's collapse that. We've used our 2 electrons and 2 protons, and now we have 2 hydrogens. Okay, we still got that oxygen. Let's add that in. Behold, it's water. So we're able to make water, and those 2 electrons that have traveled through the electron transport chain are finally combining with oxygen. So this is the step where they reach oxygen. We have, in the meantime, instead of releasing energy like a combustion reaction to go into hot, fiery heat, we've actually harnessed this. So for our 1 NADH that we input, the electrons have traveled through these complexes all the way to form water. And this actually happens extremely quickly, much more quickly than we're talking about it. Think about an electrical current, how quickly electricity flows. This is kind of similar. This is sort of an inner current. And when we're thinking about electricity, we obviously use those currents to power things in our daily lives. Similarly, we're able to use this electron current through these complexes to pump protons into the inner membrane space, ultimately arriving at 10 protons up here. And you can imagine that as this happens continuously, we're continuously feeding NADH into this cycle, this transport chain, you're really going to create a lot of protons up in this inner membrane space and really get that gradient going. Okay, so really the way to think about this is just to think about this as electron flow. It's flowing through these and we're powering this proton pump. So something you may notice here, we still have something to talk about with our complex 2. So what the heck, we completely just skipped it. We didn't need it at all. What's happening? So you may also notice something else might be missing from this electron transport chain, and that's what happened to our FADH2. So let's look more closely at complex 2. Complex 2 is specifically where FADH2 will deposit its electrons. Its other name is succinate dehydrogenase. This may sound familiar to you. Where have we heard this before? Hopefully you remember that this is step 6 from the citric acid cycle. So we actually made FADH2 in step 6, but the electrons are immediately sent from FADH2 to complex 2 and the rest of the electron transport chain. So again, this is the step where we are going to see succinate converted into fumarate, and again, the FADH2 is actually created in this step and then immediately funneled into this complex. So this is kind of interesting. It's a little bit of an M. Night Shyamalan plot twist that complex 2 is actually part of the citric acid cycle. So you can actually, if you're going back to review the citric acid cycle, kind of envision step 6, where succinate dehydrogenase is participating. Kind of just imagine that that's part of the electron transport chain. So we now have a role for complex 2. So let's go back and drop it into the chain with everything else, and let's see what's going to happen for FADH2. Okay, so notice some things here. Electrons, if you remember, FADH2 also is holding 2 electrons. So in that regard, there's no real difference between NADH and FADH2. They both hold 2 electrons. However, the difference is where they enter the chain, where their electrons are allowed to participate. So you can see that we go from complex 2 to ubiquinone to complex 3, cytochrome C, complex 4, and to water. So essentially, we're just skipping complex 1. So what does that mean? If we're thinking about protons, complex 2 didn't pump any. We skipped complex 1, so we're only going to rely on 3 and 4 to help us build up that proton gradient. So just to note, we got 10 from NADH, and we now have 6 from FADH2. Again, don't memorize that just yet. We're going to come back to it at the end. For now, let's take a closer look at what's going on at these specific redox sites within our 4 complexes and our mobile carriers. So if you remember, the whole point of cellular respiration and the electron transport chain was to harness the energy of allowing electrons to go to oxygen in a controlled way so that we can get ATP instead of releasing this as kind of a quick and dirty heat. So you can see descending free energy. Here's our free energy on the y-axis relative to allowing this to go to oxygen. So NADH transfer, you can see that this is a very gentle step. okay, in terms of we're just not allowing this to snap, just to drop all the way to 0. Flavin mononucleotide, iron sulfur. Each one of these represents a decreasing free energy. There's ubiquinone. We're also coming in from complex 2 at ubiquinone. So you can see that this is still representing a decreasing free energy. Cytochrome B, iron sulfur, cytochrome C1. Here's that second mobile carrier, cytochrome C, and then A, and then A3, and then boom, to oxygen. So that's kind of the point of doing this, or at least this is where we're able to get that current going so that we can pump protons. So I think important to think about this. This is just favorable steps so that we can control the descent and harness it to ultimately make ATP. So we found out what happens with the electrons. Once they're combined with oxygen to make water here in these protons, this is done. These are done. So we don't need this anymore. We don't need to think about it right now. This oxygen is the oxygen that you inhale. This is why we have to breathe. So we inhale oxygen so that we can do electron transport. So it's not for glycolysis. It's not directly for the citric acid cycle. It's for electron transport. And there are organisms, you think about things like sulfur-reducing bacteria. They actually can substitute sulfur for oxygen in this equation, but they have to have something to catch the electrons at the bottom. So here, oxygen is catching our electrons. If we don't have anything to catch it, this whole chain can be backed up. And we are going to end up either going through fermentation, which we'll talk about later, sort of an anaerobic metabolism, or this can result in death depending on whether the organism is able to live on those bonus fries from glycolysis. So this, again, this is what you inhale. When you're exhaling, carbon dioxide is coming from pyruvate processing in the citric acid cycle. So that's where these like larger organismal things come into play. So let's go ahead and get this ATP that we've been waiting for. So to do this, we're going to look at an enzyme called ATP synthase. So I've just got the top part highlighted here. So this is going to be the F0 or C10 or C-ring subunit. So C10, if it's got 10 parts, different organisms can have this arranged a little bit differently. So I'll also refer to this as C-ring. So here's our F0 component. You can see that it's embedded in the inner membrane. So down here, still the matrix. Up here, the intermembrane space, and you can see some business about to go down with this proton. So we know that we've built up a pretty sturdy proton gradient. So we've got a lot of protons up here. The concentration is lower in the matrix. So that's setting us up for getting that ATP. So this C-ring or F0 subunit has spots for binding protons. So protons enter the F0 subunit from the intermembrane space, and they bind specifically to these sites. As protons bind these sites, I'm going to just mark this one as kind of a darker orange so that we can track it. It causes this complex to spin. So you can see it starts to spin. We're going to go in this direction. And it, of course, happens much faster than this, but just so you can track it. And eventually, we're going to go all the way around. And these protons will exit from the back into the matrix. So that's it. They waited in line. They did their Schlitterbahn ride. And now they're back out. And they're free to do whatever they want. They can go over to complex 4 and combine to make water. They can go back through these proton pumps and go again. So there's just a lot of things that can happen. We don't need to think too specifically about this. It did its job and it helped to turn this wheel. So we just tracked one proton, but really as that one proton went around, it would leave vacant spots behind it, and there would be protons filling all of these spots, causing this subunit to continuously spin. We'd have kind of a consistent flow of protons back into the matrix. So think about this as sort of like a water wheel. If there's a lot of water pressure, turning a wheel and then coming back out. That's exactly what's happening here, except we're using protons. So once that's exited from this subunit, we've caused this to spin, we can then start to see how we're going to get our ATP. So again, imagine this top subunit is continuously spinning. That brings us to the bottom subunit. Also, we call this the F1 subunit. This part is going to stick out into the matrix. So as we're spinning from the top subunit here, our F1 subunit sticking out into the matrix, it's going to cause part of this bottom subunit to spin as well. So this kind of like internal ring, this sort of internal ring in here will start to spin inside this larger complex. It's going to be composed of alpha and beta components. So we can just think about this sort of stalk spinning within this ring complex. So the F1 part, we're sticking out. We've got alpha and beta subunits, and these are going to catalyze the creation of ATP from ADP. So let's see how that happens. So as again, everything's spinning, the energy of the spin is used to take an inorganic phosphate and stick it onto ADP, remembering that that's not a favorable thing to happen because all of our phosphates are negatively charged. ADP has 2 phosphates. We add a third one on, all of these negative charges together. That isn't something that naturally wants to happen, so we have to use energy to create it. And this is called oxidative phosphorylation. So one thing you might be wondering is exactly how many protons you have to put into this mechanism to spin this enough to create our 1 ATP. So it takes approximately 4 protons. Some people kind of think it might be between 3 and 4, but we're going to use 4 because I like the way that it makes the math work out. So it takes approximately 4 protons going around this C ring, this F0 subunit, to spin our F1 subunit enough to make 1 ATP. So the conversion there is 4 protons make 1 ATP. So let's see how this math works out. If it takes 4 protons to make 1 ATP, how many ATP can we get from 1 NADH? So we know as we deposited electrons from NADH into the electron transport chain, we pumped 10 protons. So 10 protons that we got from NADH, pumping 1 NADH's worth of electrons, divided by the 4 protons per ATP, this is how many it takes to make an ATP, this gives us 2.5. Therefore, we get 2.5 ATP per NADH. Whoa, so we can actually quantitate this. So let's get ready to get that 32 to 36 ATP. So what about F-ADH2? We know that we pumped 6 protons from depositing the electrons on F-ADH2 at complex 2. So these 6 protons from F-ADH2, again, it takes 4 protons to make an ATP. This gives us 1.5. So therefore, we get 1.5 ATP per F-ADH2. So even though NAD and F-ADH2 hold 2 electrons, we get 2.5 ATP from NADH, but only 1.5 per F-ADH2. So let's start adding these totals up. So from glycolysis, we know that we got 10 NADH. From glycolysis, pyruvate processing, and the citric acid cycle, we got 10 NADH. This is going to give us 2.5 ATP each. That will give us, per glucose, 25 ATP. So remember again, the 1080H came from glycolysis, pyruvate processing, and the citric acid cycle. For every 1 NADH, we get 2.5 ATP, so we just multiply that by 10 and get 25 ATP. We got 2 FADH2, which only give us 1.5 ATP each. So since we had 2 of those, we get 3 ATP, specifically from FADH2. So far, that's 28 ATP. So that's not our 32 to 36, but don't forget about those 2 ATP from glycolysis, those bonus fries, and the 2 ATP/GTP from step 5 of the citric acid cycle. If we add those to our 28, that will give us 32 net ATP. So from oxidative phosphorylation, which happened only using ATP synthase after electron transport, we got a net 28 ATP. We added back the 2 from glycolysis and the 2 from the citric acid cycle to give us that 32 net ATP. If you're wondering about the 36 possible ATP, you can arrive at the 36 if you use a different number of protons per ATP. So I like 4 because it works out really well, but if you want to get the 36, you can pretend that it takes a little bit less than 4. So we did it. We have calculated now how we actually get those ATP. We have seen every step at how we arrive at that number. So let's also just recap by remembering our cellular respiration reaction. So glucose, we saw that at the beginning. That was our original input into glycolysis. We saw the carbon dioxide leave at pyruvate processing and the citric acid cycle. Oxygen was used to catch electrons. Some of this water became from forming water as these electrons are captured at the end of complex 4. The energy, we are now seeing that payoff that we got from oxidative phosphorylation. So we got all of the ATP. So this has been kind of a long journey, but cellular respiration is one of the coolest biochemical wonders that can happen in the cell. If you think about it, it's really just taking carbon from the things that we eat and taking electrons from it to make effectively a tiny little current inside your mitochondria. Will power this proton pump that will allow us to have ATP synthase create ATP. So this is actually a very cool thing. It's easy to make this really hard and complicated because there are a lot of structures and a lot of new names. But really, just think about the pathway the electrons take. They go from carbon to oxygen, combine that to make water. That's really the gist of it. There's some other things along the way, but that's going to be our main point. So hopefully you guys feel good with cellular respiration. And in the next set of videos, we'll be starting chapter 10, which will give us the reverse. We'll be looking at photosynthesis and how do we get those electrons from oxygen back onto our carbon, so sort of recharging those batteries.